today I'm mainly going to focus it's here, yeah? On sort of the technique of NMR and how we have tried to move that into the future. So my main point is that you probably, most of you have heard about NMR, probably seen NMR spectra definitely in the previous talk. But most likely, very, very few of you have handled NMR data. And potentially very few of your labs have produced any of the 100 samples that John talked about. So that's what I am trying to change. My goal is to make NMR a bit more available. I'm clearly biased, but I think it is a tremendous technique. We have potential to see dynamics that no other technique has. Yet, some of these experiments are probably only available to maybe a few handful of people in the world. And I don't think it's fair. I have probably been a part of that myself, having sort of developed some of those experiments and going back and try to analyze my data from my postdoc time. I realized that this is not as easy as I thought it was. So what we are doing, we're using AI and deep learning for two things. We are using it to improve the sensitivity of NMR, as you probably know and also heard in the previous talk. NMR is not very sensitive. Secondly, we are trying to lower the criteria. That means the criteria for labeling. Most of advanced NMR would require you to make samples in D2O. That's not only expensive, but also many cells, mammalian cells, do not grow on D2O. And thirdly, we are trying to make the analysis easier. We are trying to make it sort of a semi-autonomous, which means that it should be as easy as running an AlphaFold prediction. So as much as probably very few of you had ever sort of simulated or predicted a protein structure prior to AlphaFold 2, it is my goal that in the future, most of us and you and future students would actually be able to handle NMR data themselves without needing to establish a collaboration with the two handful of people around the world. So what is deep learning? You might have heard a lot about it. You definitely used it either on your iPhone or your other phone or somewhere else. It's sort of mathematically very simple. It's just a mapping. It's just a mapping from a given input to a desired output. Now, this input and output can be in various domains. It can be from text to pictures. That's one of the examples, would be called as a DALL-E. You type a text and it spits out a picture. It can be from text to text, so ChatGPT. It can be from picture to text, which potentially would be facial recognition, etc., etc. Now, how this works is that we typically have a set of layers, we call them, where we pass the information through, and each layer here will have a linear transformation given by this tensor big A. That will be followed by a nonlinear transformation, and the reason for that is that we have the universal approximation theorem, which tells you that we can approximate any function with a sufficient large number of layers. So what we need to do is we need to make that mapping do what we want, and that's what we call training. Training effectively just means that we need to optimize all these big A's here, all the parameters within the big A's, which someone refers to as weights, so that the mapping do what we want. Now that requires lots of data, and that is by far the biggest challenge of most AI and machine learning projects, simply to finding training data. Many of you will have been online, you will be asked, is this a cow or a bridge or a motorcycle, whatever, and you have to click in order to do whatever you like to do. Well, any AI can do that. So it's just because someone wants training data in the back. Now, so we need billions of training data. What we have with NMR that actually probably not many other fields have is that based on the very in-depth understanding of the theory and of many years of my forefathers in the field have spent on understanding these experiments in such great details, it means that we can actually simulate NMR data at a level where no one can distinguish them from what comes out of the spectrometer. Now that was fun in the past because we could do least squares fitting, but if we turn it upside down on its head, it actually means that we are able to simulate an unlimited amount of realistic training data. Okay, so that means this biggest bottleneck that there are in most fields of AI, we already passed that. So the only thing, what we do need to really consider, and I'm going to spend quite a bit of time in this talk talking about that, is that of AI hallucinating, AI making up things that are not true. AI just generating things that might look right, but it's not. And one of the things that are required is really that the mapping we are looking for needs to exist. AI cannot generate magic. Well, it can, but then we don't want to use it. So the mapping from the input to the output needs to exist. And that means if I want to use AI to, let's say I have an NMR spectrum that looks crowded and bad and overlapped and everything into a nice high resolution map, I need to make sure that mapping actually mathematically exists. Okay, so what we are going to do is that instead of just passing in so-called normal NMR data, we are going to create additional dimensions in the NMR spectra that's not for me or any other of your favorite NMR spectroscopists to look at. No, but that is to compress additional information into the NMR data that the AI likes to look at. So orthogonal mathematical information. So let's take a look back. How does NMR work? Pre-deep learning. It's a very linear process. We'll go to the spectrometer, we record some spectra using a pulse sequence, it's called. Then we do a Fourier transform, we look at the spectra, we do least squares fit, and then some analysis. So over the last few years, what has happened is that some of these errors have been swapped with potential use of AI and deep learning. And that has improved each of these steps. Now, if we really want to change NMR, what we need to do is we need to combine the entire process. As I say, we need to make sure that the data that is generated in the spectrometer is not something that's for me. No, it's something that is geared for the AI to analyze. Okay? So what we really need to think about, as I say again, is really to make sure that the neural network is not hallucinating. And you might think that DALL-E and ChatGPT are super amazing. They are for sure. But if you think about it, you go to DALL-E, you write in the prompt, I want to see a pink elephant jump on the top of a mountain. It gives you a picture of a pink elephant jumping on top of a mountain. And you're super excited, right? The thing is, it can be any pink elephant on any mountain, jumping in any way or sort it may like, and it has fulfilled your criteria. Our networks are put under much higher constraint. So we are not asking our neural network to just generate an NMR spectrum. No, we're asking the neural network to generate the NMR spectrum of the protein that was in the magnet. And as you can imagine, that is a much harder constraint. So what we need to make sure is that what comes out is something we can evaluate, we can feel good about it. And we are scientists, so what do we want? We want uncertainties. Since our very early days, at least my old professor was always telling me that any number without an error bound and uncertainty means nothing. So that's what we're going to do. We're training our neural network not only to give us a result, for example, an enhanced spectrum, but everything comes along with an uncertainty, okay, which we can judge. Essentially, it has to be a normal distributed and follow the other criteria. So let me show you a few examples of things that are possible and not possible, and how we circumvent them, and how we generate these additional data and additional dimensions in NMR data. So on the bottom, I showed you two spectra. one to the left and one to the right. Now, most of you have seen NMR of small molecules. We have what's called a triplet if a proton is coupled to two other protons. So this, you may say, oh, it's a triplet. I recognize the pattern. But it could as well be three singlets that just by accident are placed next to each other. So that means neither me nor any AI or deep learning neural network for that sake is able to tell you or able to decouple these ones into a singlet. There's simply not enough information. So what we can do, we can evolve the scalar coupling, so to speak, and then it becomes very clear that the one to the right is indeed originating from a true triplet, because you see the scalar coupling evolves, whereas the left one is just composed of three singlets. So these are sort of the tricks we're going to use with NMR. As I say, we cannot create magic, but we can provide additional information into the data. So let's see how we're using this and trying to make life a bit easier. So what we are aiming at is to lower the criteria for samples. We want them to be able to be grown in just H2O, normal water, from the tap or your molecule, and just simple labeling, uniform C13, without any specific isotopes. That means, first of all, it becomes much cheaper, and secondly, you can grow samples in mammalian cells. Okay. The problem is that when we look at spectra, so this is a small H, 50 kilodalton chaperone, ATNHAB1, so like a HSP40, and you will see substantial overlap. So effectively, those spectra are not possible without any labeling. We have an aromatic spectrum and also lots of overlap, so that's also really not possible currently. There are techniques such as constant time and specific labeling. However, those techniques are definitely not applicable to aromatic regions because of various scalar couplings and various effects. So what we've tried to do is, can we take these pancake spectra and can we transform them into look like something that was produced with an expensive labeling scheme? So what we're going to do, we learned previously that, well, I can change the scalar coupling. I can modulate that as we saw in the 1D spectra. So here we are simply modulating it in a 2D spectrum. And these are now 1D slices from a 2D spectrum. And that means having these additional coupling evolutions, we are able to see if those peaks originate from a singlet like the red one, here like two singlets, or a doublet, which would be the green one. Same with a triplet or a doublet. And that means we will be able to go ahead and decouple these spectra, make the peaks much, much sharper. What our neural network does, and I won't give you all the details of training, you can read about it in our recent papers, is that it will then take two spectra, one without coupling evolution and one where we have these funky shapes, and it will transform it into a decoupled spectra and a map of the uncertainty. So we get uncertainty on a point-by-point basis. We can show that this uncertainty is indeed very Gaussian distributed, and the RMSD matches exactly what we expect from what is predicted. So we here have a neural network that can enhance the resolution of the spectra, and not only that, but we have a neural network that actually knows when it fails. It increases the uncertainty. So I think it is fair that neural networks have their limitations as long as they would tell the user when they fail and when they encounter something challenging. This neural network does it by increasing the uncertainty. So let's look at the experimental data we had before. Remember, this is a 50 kilodalton HSP40 chaperone, which is C-terminal domain. And what you will see is that these very overlapped regions are now transformed into something that looks like it was made with a specific isotope labels. Very much higher resolution. If we take a slice through it, you will see the resolution enhancement is quite significant. And not unsurprisingly, regions that we do expect to be very challenging, like this sort of big pancake, is where you will see a larger uncertainty that's shown in the back with the blue. So the neural network clearly knows what it can do well and what it cannot do well. Yet the resolution enhancement is definitely quite substantial. Let's move a bit bigger. This one here is an 80 kilodalton valence synthase G, so a single chain. Again, left spectrum, no specific labeling, just grown in H2O, nothing special, and you see as you would expect, it's very broad and not very easy to analyze. The neural network makes this one actually very high resolution where you potentially could see binding or any other things following any of these peaks. And again, now you may ask yourself, this does look a bit like magic, doesn't it? Is it true? I tell you we have uncertainties, but can we really believe in those? So, for this protein, we are very fortunate. We can actually go and make it in bacterial cells. We can make it in E. coli. We can actually deuterate it. We can use these expensive precursors and specific isotope labeling. And we can now compare how does the protein look in blue if it was made in D2O with this uniformly deuterated sample with specifically labeled precursors. And you see all the blue dots match exactly the red dots. The red dots is what came out of the neural network. So indeed, yeah, the neural network does produce not a spectrum, but actually the spectrum that we are looking for. We can go bigger. This is a 360 kilodalton protein, and again, this is now getting even worse. And you see the resolution enhancement is quite substantial. We can do the same test again, zooming into the worst region. And again, you see effectively every blue dot has a red peak next to it. All honesty, I think 360 kilodalton is currently our limit. So if you do want to go to an order of magnitude higher ribosome you heard in the previous talk, you're probably still needing to deuterate. But I think the limits have been pushed quite far here, at least an order of magnitude. So this is methyl groups. There are definitely things that are as fun, if not more fun, and I think it's the aromatic residues. Those are tremendously very, very difficult to probe with NMR, and you see here a spectrum of just a 20 kilodalton protein, and apart from these tryptophans up here, these also tryptophans, it's really hard to extract any information. For aromatics, there are a few labeling schemes, but they're very tricky, not very high yield proteins, and other techniques are really not possible, such as constant time, etc. We did train the neural network similarly as we did for the methyl groups to transform the spectra of aromatics residues. And here's one example. As I say, this is probably our limit currently. This is again the 50 kilodalton chaperone. You see the spectrum prior to our neural network is not usable in any shape or form. However, once it's transformed, you see we have very high resolution. We can make slices through, and you see it's both very accurate and very high resolution. So this was the first step. How can we improve the spectral quality? How can we make it easier for people to use NMR without requiring all these specific labelings? The next step is how can we analyze NMR data? We're not fully there yet, but taking a few steps along the way. And I think some of the hardest things to analyze is sort of protein dynamics. It's also where NMR can actually really contribute and is totally complementary to cryo-EM and other structural techniques. On the back of high resolution structures, having this dynamics is a very valuable thing. Problem is analyzing these data. So for example, analyzing a protein going between various conformations, sampling various states, extracting rates of that, extracting populations is by far trivial. And there you might have heard of these techniques, CPMG, DIST or CEST, which is like chemical exchange saturation, or structure and dynamics of these states is really not that simple. So what it requires is probably 10, 20 years of experience. It needs you to be very good at guessing starting parameters for your least squares fit. And in the end, it definitely requires an expert to judge if your analysis is valid or not. So how started that is we took initially one of the very challenging so-called CEST experiment, the chemical exchange, which are curves that looks like these blue one here in the background. And what we did, we trained a neural network to find out the chemical shifts of the ground state and this low populated excited states. Fitting these data with least squares fit is probably just, as I say, 5, 10 people in the world who can do it. We teamed up with the person who wrote the program, and we ran our neural network through the data, and then in the evening did a blind test with him. And it all matched perfectly. These numbers here, see, they all have uncertainties as well. And the interesting bit, I'm not showing the plot here, but the agreement was perfectly within this. So the RMSD between our neural network and his least squares fit was effectively the uncertainties predicted here. So it's a one-shot analysis. There's one line in a terminal. We might implement a button on the spectrometer, but effectively one-shot analysis. Now, these are only providing you with the chemical shift, and as you know or heard before, getting really the thermodynamics, getting population, getting rates, getting kinetics is rather interesting as well. So, what we have done, how long do I have? Okay. What we have done is moved forward. This is not published, and I want your input, which is why I'm trying something I only tried once before. So we're going to try and do a live demonstration to see how hard or how easy it is. So effectively, the other set of experiments is CEST and nitrogens. You see they have different dips, which indicate that you have a type of dynamics. You have a ground state and a low populated state. The challenge is with these experiments to extract out both the kinetics and the thermodynamics. You need to use fairly advanced software. You need to transform into various file formats. You need to fit the data, and you need to be able to judge the quality of the fit. So we have trained a neural network to do that, and as I said, I only tried it once. Let's see if this is working. So this is my computer in London, so it's not too far. So let's hope it's working. So what we're going to do, I have my friend in India record 2 sets of data on this FF domain that's folding and shuttling between folded and unfolded state. So first we need the mouse to work. Without that, it's not good. I'll maybe use the trackpad. So first, if you have used Bruker spectrometers, put up your hand. Have you seen a Bruker spectrometer? Yeah, that's a lot. Good. So this one just requires it to sit on the spectrometer and someone tell you where the data would be. So first we need 2 data sets. As I said, like with the couplings, we have, in this case, a low B1 and a high B1. The key is to have 2 sets of data so the neural network can extract the information underneath. So this is on the spectrometer, go to data. This is a low one, low B1, into, you see the Bruker structure, those who knows it, P data. And I click 1, and I think I just say yes. Let's see if this works. Yeah, so there's a spectrum. So far, so good. Remember, I need to read in the other one too. This is a high B1. And this is, there's no cheating here, so if it fails, it fails. I do have a pre-recorded version, but I thought that would be cheating. So these are now the second data set we recorded on top. And my mouse is out of battery. Here. So then we need to load a peak list. And what normally would happen when you load a peak list is you get it from a friend, and of course it doesn't fit. Right. It's not like when you get a structure from a crystallography, it sort of normally fits in the density. So we will just sort of move the peaks here. You can see you can move them until you think it fits. I think this is pretty good. Yeah? So, what region shall we take? Maria, what do you say? Just to show that it's not planned. Or someone else.
What do you want me to take?
Oh, I can pick a region.
122.
Where's 122? Where's 122? Oh, 122 ppm. So here, that one. Okay. Sorry if I run out of time. So then we just zoom in on this 122, so this region here, yeah? What we will first do, we will just click on the peak. What we need to do is we will need to, sorry, it's hard to see here, fit the peak. We'll need to fit the intensity. So we click on it, and then we'll sort of judge if that looks good. I think the red dots is fairly close to the blue line, yeah? Do you agree on that? So I close, and now it's ready to do CEST. The first time it's going to take quite a while. Let me click, then I can talk while it's thinking. Because the very first time, remember, as I said, these A's, the layers, the parameters, I have billions and billions of those. It will be gigabytes and gigabytes. So the first click, what's going to happen is that my poor computer in the hot UCL office is now scooping all these weights onto the GPU. It's arranging the neural network and it's setting it all up. And the first click should typically take a half a minute, so I'll speak a bit slower. Yeah, there. So it has now analyzed everything. It was one click. So it has predicted the rate, the population, the chemical shift, the relaxation rates, etc., etc. And it took the data straight off the spectrometer. It all comes with uncertainties, and I'll show in a few slides. This is all, of course, validated uncertainties. So let me take one more. Who wants to choose another peak? Why don't you choose one, Paula? You sit on the first row. Can you see it? Which one? This one? No. Yeah. This? 12. That one. Okay. Sorry. I have some that actually shows a big CEST response. So see, remember first we have the peak fit. One that actually shows the two dips. The two dips are easier to fit. Even like a first-year student or my 5-year-old daughter can actually see where the dips are by eye. The trickier one, see, now we see, do you want to count how many seconds? I click now. 1, 2, 3, oh, yeah. So it's already analyzed. So now it can analyze all that. We need to find one with a few more dips, don't we? I think this one here. That's in where there's a lot of folding. What this network does, which I'm not sure we will find an example, what it is doing is that if some of these data look bad, it will actually tell you that the data are out of scope, and it will refuse to analyze it for you. I'm not sure if I have time to find an example. See, here was one with 2 dips, and what you again see is that all the populations and the rates are very, very similar. So we hope that, I see Maria standing up, that means she's about to finish. So no more peaks analyzed, but I think we showed that it's not impossible to use these networks, and we really hope that more and more will be able to analyze them. Of course, this still requires it to be set up at the spectrometer and things recorded, but I think analysis, we're getting very close to having that nailed. So the point is, as I say, this neural network is what's called an autoencoder. It goes forward and backwards. So we have the spectral parameter you saw after I took the data and ran the neural network. What the network is doing in the back is that it's actually looping it back. And if the back calculation doesn't look like what came in, that means that the parameters are not trustworthy, and it will give you a big message saying it's out of scope, and it will refuse to analyze it any further. You can then either email me, or you can ask your other colleague, or you can just ignore the data. But at least you will not have any false data in there. So these are a few comparisons with what we get from least squares fitting. You can see it's a perfect straight line, so definitely we can trust these. Let me go just to the end. I think we now have tools available to do autonomous analysis. We have tools available to lower the barrier of entrance to NMR by having less strict labeling schemes. And we are still moving on in both of these areas. Of course, as you can imagine, quite a few people. This was started by very talented postdoc called Gogeland, And then we have collaborators with Rina, works on the chaperones, quite challenging, promote India. And then this is already implemented in CCPN, part of it, the NMR software, and then some people for giving us money. And thank you very much. Oh, yeah.